Hello everyone. I will introduce you to the research project of my PhD, which focus on how dendrites supports the formation and reactivation of sequential memories. So first, I would like to make some background so that we can better understand the relevance of the problem at stake. Well, we focus when we talk about network memories on the concept of cell assemblies. Cell assembly, indeed, since the seminal work of Daniel Hebb in 1949, are considered the substrate of associative memories. And cell assemblies are formed through the repeated activation of two or more cells which tend to become associated to each other through Hebbian plasticity. Now, these stable distribution are formed through the repeated presentation of a stimulus. For example, how is shown in this illustration by Joachim Fuster of a network memory. Vision and touch are co-activated, and this leads to the reactivation of vision when the stimulus through touch is presented. Now, importantly, these cell assemblies has also been validated as a paradigm by computational work. In particular, the work of Lidwin Kumar and Doiron in 2014 show that cell assemblies can be effectively formed through spike-time dependent plasticity, and that these engrams, which are formed by the repeated coactivation of the cells, are robust to noise and are maintained over time. Now, it seems that kind of cell assembly works very well to represent memories. The problem is that some memories are more complicated. In particular, as Gallistel points out, most of memories we have about the real world are not in themselves association, but are rather relationships, in the sense that we need to know in which relationships objects are in reality to understand the world rather than just associate them. And this is a particular case is in language, and in one of the most basic functions of language, word recognition. In particular, words in word recognition have to be considered as temporally structured memories, in which the same features, like the three letters D, O, and G, can appear in one or the other sense. And the brain, humans in general, have to be able to distinguish this to stimulus presentation in order to reactivate the correct word memories. So this problem we also can call sequence labeling, in the sense that we have a sequence of three phonemes, D, O, and G, and we want to recognize whether it belongs to the dog or God memory. Now, the problem that we lack temporal structure into associative memories is well known in cognitive neuroscience. Indeed, most models, like trace here, use a technique to disentangle between these two memories, which is to diagonalize or to create an orthogonal dimension of the onset time of the phoneme with respect to its identity, so that the feature D, which is presented in dog and God, is are effectively two different features, which is D in position 1 and D in position 3. We do know, though, that this is an implausible mechanism, because our brain cannot kind of know which is the order of the phonemes in advance. Now, coming back to spiking neural network, we also would like to point out that this problem is not solved in spiking neural network in general, in the sense that despite there are several models that address sequence recognition and sequence labeling, none of these models use the cell assembly paradigm to store this memory. And for example, we have a very important work, which is the reservoir computing framework, that use hidden neural states, and these hidden neural states can indeed recognize sequence like forward and reverse, but they do not use cell assemblies. Or either forced learning, which kind of use the feedback connectivity to recognize a sequence and to reproduce a sequence. Or other algorithms like modern eProp or SuperSpike from Zenk and Bellek, or more old work like Gudig and Sopolinsky, which can effectively recognize sequence of spikes or sequences of inputs, but they use supervised plasticity. And also in this case, there is a readout at the end of the network and is not a specific word population which is reactivated, nor a cell assembly. Now, we think that to introduce, let's say, reconciliate the cell assembly paradigm and the spiking neural network framework, we should consider an important element of biology, which are dendrites. Indeed, we know now for a couple of decades very well that dendrites increase the computational power of neuron models. In particular, they allow the neuron to exploit the spatial distribution of synapses. Three elements we consider are crucial of this specialization of, let's say, memories. One is that the dendrites make the neuron computationally equivalent to a multiple layer network, which allows for integration before the soma. In addition, these segregated compartments also allow for the formation of independent memories, which can maintain across time, and they do not interfere with each other, like was showed by Bonen-Klopath and Castellaki's work. Eventually, there is a third result, which is slightly less known to the general literature, which is the temporal extent that dendrites add to neuronal activation. And in particular, the fact that through NMDA receptors, we can extend the integration time of the neuron. We investigated this problem or these features in a specific study which was published a couple of years ago, and we study how a simple neuron model with two segregated dendritic compartments, which are equipped with NMDA receptor, can produce an temporally extended depolarization in the soma and in the dendrites first, in response to an increasing synaptic input, which means that the stronger the input the dendritic compartment receives, the longer lasts the depolarization. And this happens because of the NMDA nonlinearity, which lasts long in time. Now, given this property, we think that dendrites can actually help for the formation of memory with sequential structure. So I will now discuss the network model we investigated and start with describing it in its methods. We created a network with 2000 tripod neurons, that is the model I just introduced, and 500 inhibitory interneurons. These inhibitory neurons are divided in slow spiking that target the dendrites and fast spiking that target the soma. And this model has been compared with two traditional network models that also can process associative memories or can process sequential stimuli, like the Lidwin-Kummer-Endeuron and Duarte-Morrison model. Now, in addition, we created a set of word and phoneme projections that last each 50 milliseconds. The phonemes and the words last 50 milliseconds times the words times the phoneme in the words. Now, each stimulus targets a randomly selected assembly of 5% of the network neurons, the excitatory neurons, target the dendrites, of course. Now, the idea is that during a first phase in which words and phonemes are co-activated, there will be created association between the phonemes and the words. And this association can be then leveraged to reactivate the words during a recall phase. Now, the associations are formed through unsupervised plasticity rule, which are biologically plausible. One of these is the plasticity rule that applies to dendrites and is a voltage-based spike time dependent plasticity rule. This voltage-based plasticity rule allows for the formation of strong pre to postsynaptic connections between the presynaptic firing neuron and the postsynaptic dendrite. In addition, we also applies multiplicative synaptic scaling that helps the neurons to prevent runaway excitation problems. The inhibitory neurons also undergo a plasticity rule, which belongs to the, let's say, category of homeostatic inhibition, in the sense that this plasticity rule, like the one proposed by Vogels, tends to maintain the neuron at specific level, which are defined by the target parameter of the plasticity rule. We split this plasticity rule in two, one for the somatic and one for the dendritic compartments. And for the somatic compartment, we target the firing rate of the neuron through the fast spiking neurons, and for the dendritic compartment, we target the membrane potential of the dendritic compartment, maintaining it at minus 70 millivolts. Now, the inputs we use for this network are a selection of vocabularies. Here we have a sample of 5. These vocabularies have an increasing complexity in terms of overlap between the phoneme that we call phonological overlap. This overlap, for example, here we see in the overlap vocabulary, we see that there are many words that kind of look alike, there are less phonemes than words, and some of the words are contained in other words. So this means that in order to recognize which word was presented, the network has to be able to recognize the order and also to recognize when words were contained in other words. Now, let's look at the results of the network. First, we notice that the spiking pattern is asynchronous and has peaks in the spectrum at the frequencies with which the inputs were presented, like 20 milliseconds for the phonemes and 5 to 8 or 12 Hz for the words. Now, most importantly, we see that during the recall phase, which is when only phonemes are activated, upon the presentation of phonemes, also word populations are reactivated. And we see that kind of these populations are reactivated selectively, dependently on which phonemes were presented. Now, in order to measure the reactivation of this word population and whether it's effective, in the sense whether these memory are correctly recognized, we devised a simple method which consists in measuring the firing rate of the cell assembly. Now, in addition, we also shift the interval in which the firing rate is measured, because we know from preliminary results that this delay is important, in the sense that the maximum of activity of the phoneme of the word assembly happens with a certain delay from the initial presentation of the word inputs. And in particular, we had sampled at around 100 milliseconds after the first phoneme is presented. We see that we can measure the target population, the rate of the target population, and that this rate is systematically larger than the rate of other words. Like here we measure the rate of Paul and we compare it with the rate of 4 words with several overlapping phonemes. Now, it's important also to notice that in this approach we have no train readout, which means that it is the activity of the cell assembly and not the hidden network activity, nor a state that must be filtered through a readout that represents the activation of the memory. Now, let's have a more systematic look, and we consider the word which has the strongest activation for each word presentation. Okay, so the cell assembly with the strong activation for each word presentation. When we take this route, we can construct a confusion matrix in which we have the target input and the reactivated population. We see that the best score, or kind of the most correct activation, doesn't take place at the beginning of the word, but it happens with 100 milliseconds after the offset of the word. And this we can also see from more, like, let's say, static analysis, in which we compute for every delay, we compute the trace of the matrix and compute the Cohen K-score, which takes care of removing chance level results. So we see that when comparing vocabularies, in general, we have that only dendritic networks recognize overlapping vocabularies, which means vocabularies with shared phonemes. And we see that single compartment model like the Lidwin-Kumar and Duarte model can recognize vocabularies with only identity or no overlap among the words, but they progressively fail when the overlap among words increase. Indeed, we also see that the word overlap or phonological overlap is the most critical variable to predict the Cohen K-score of the model. Now, to understand how this works, we have to look at the formation of network memories. And this formation can be, let's say, followed by taking the average of the phoneme towards population synaptic strength, which means here we take all the neurons that belong to a phoneme assembly and we take the average of connection between these neurons to the word neurons. We see that in the tripod network, but only in the point neuron, these feed-forward connections are strong and are comparable to the recurrent associative memories. And this happens through time, slowly and more regularly for dendrites, which indicates that dendritic learning is lower but also more stable. Now, when we look at this structure across all the lexica, across all the vocabularies, we see that in general for an increasing overlap between the phonemes belonging to the vocabulary, we have more and more confused feed-forward connections. And this is perhaps the cause of the decreased accuracy. Indeed, we see, looking more in the depth of one specific lexicon, we see that the connection between the phonemes and the words have an internal structure, in the sense that for an augmenting phoneme index, for increasing phoneme index, for example, this is the uniqueness point of the word, which means the point in which the word can be recognized, the phonemes have increasing strength onto the word. And this structure within the synaptic weights is actually necessary for correct recognition. Indeed, we tested that if we flatten the structure, then the capacity of the network to recognize the vocabularies decrease. And this happens the most for vocabularies with large phonetic overlap. In conclusion, we also look at which is the critical variable that allows for this mechanism to take place. And in particular, we see that the critical variable is the target voltage of the inhibitory spike time dependent plasticity. Indeed, let's look at how the non-linearity of the network has an effect on the recognition. We see that when we decrease the NMDA to AMPA ratio and we decrease the NMDA decay time scale, also recognition go down, although quite slowly, in the sense that still the minimum, the performance loss is less than 60%. But when we play around with dendritic inhibition, which is the plasticity mechanism that kind of target the dendritic potential of the tripod neuron, we see that the performance loss reach the 80%, which means the neuron cannot be, the network cannot process network memories at all. And this can be also seen systematically through comparison of the NMDA and the dendritic inhibition target potential. In conclusion, we see that NMDA receptors in segregated dendrites endue the neuron with short-term dendritic memory. And because of this dendritic depolarization, phonemes and word stimuli last longer in the dendritic network and can establish connection through spike time dependent plasticity, with no competition between words and phonemes towards connections. So Because of this mechanism, there is eventually spontaneous reactivation of word assemblies following the phonemic inputs, and the critical mechanism to make this recognition possible is inhibitory control of the NMDA nonlinearity.